Every year, more than 70% of Canadian youth between the ages of 12 to 17 report being bullied at least once. Bullying is repeated, intentional behavior with an obvious power imbalance where the bully exerts power over the victim. This can take the form of physical harm, social exclusion, online harassment, or verbal harassment, which is the most common form of bullying. Adolescence is a critical time for brain development, and exposure to bullying during these years can disrupt emotional, social, and cognitive growth. And what we see in existing literature is that youth who are bullied or peer victimized are nearly 3 times more likely to report symptoms of depression, and this depression tends to last into adulthood. But here's where we run into our challenge. Not every child who experiences peer victimization develops depression. While the risk is higher, the majority of young people who are peer victimized do not go on to receive a depression diagnosis. This suggests to us that there are individual differences making some kids more resilient, while others are more vulnerable to depression after peer victimization. This idea fits with something called the differential susceptibility hypothesis. Simply put, it proposes that some individuals are more sensitive to their environments for better or for worse, and that sensitivity might be influenced by genetics. Given this consideration, we turned our attention to a gene called tryptophan hydroxylase 1, or TPH1 for short. TPH1 is important because it helps the body produce serotonin, which is a brain chemical and hormone involved in mood regulation. Small genetic differences, called single-nucleotide polymorphisms, or SNPs, can alter how this gene functions, and these variations have been linked to how people respond to antidepressants and in some studies to how childhood adversity later influences behaviors. That made it a promising candidate for a current investigation. So hi, my name is Allison. I worked with Dr. Paul Arnold's lab this past summer with funding from Peer, and we looked at gene by environment interactions in adolescent depression, in particular the role of TPH1 in peer victimization. That being how different variations of TPH1 interact with peer victimization, leading to different levels of depressive symptoms. We had 3 aims for our study. The first was to assess whether specific single-nucleotide polymorphisms in TPH1 uniquely predict depressive symptoms. Second, to evaluate whether TPH1 variants moderate the relationship between peer victimization and depression using gene-by-environment interaction frameworks, which look at how genes interact with our environment to create different outcomes. Finally, we wanted to test whether polygenic risk scores, a score that summarizes the risk of depression based on individuals' unique genes, improve prediction. To explore this, we used data from the McMaster Teen Study, which followed nearly 900 children starting at age 10. These youth were assessed yearly, and as adults, they provided saliva samples for genetic testing. For our project, we focused on 298 of these participants who completed surveys on bullying and depression and also had available DNA samples. We saw a good distribution of gender and a greater number of children reporting peer victimization. Using genetic tools, we identified 7 specific single-nucleotide polymorphisms within the TPH1 gene, of which 5 were retained due to lack of variation in the other 2. And from here we ran the following analyses: hierarchical regression, which examines variance in depression scores accounted for by different factors, such as demographics, peer victimization, and individual single-nucleotide polymorphisms. Linear mixed models, which examine differences in depression scores between different genotypes, where we found that victimization-absent participants with heterozygous genotypes had lower depression scores. And polygenic risk score, which aims to calculate genetic risks associated with disease, and for which we did not find significant genetic risk for depression in our sample. However, this test is highly dependent on sample size. But what is this actually telling us? So first, consistent with past research, youth from lower income families, females, and those who experience bullying reported higher levels of depressive symptoms. Second, one particular variation called RS21107 showed a small but significant association with depressive symptoms, indicating potential predictive power of this genetic variant. Third, and perhaps most interesting, when we look at gene by environment interactions, we found evidence that certain genetic variations moderated the effects of bullying. Essentially, this is telling us that depression scores were not consistent between our different genotypes. Those genotypes being the reference with 2 copies of the most common gene, heterozygous with 1 copy of the reference and 1 different one, and alternate, which is 2 copies of the different gene. And we were seeing that young people with heterozygous genotypes carrying 1 copy of each genetic variant who had not previously experienced bullying tended to have fewer depressive symptoms than other groups, suggesting that under the right environmental conditions, certain genetic profiles may be protective. Now, this does not mean that there is a better genetic profile when it comes to bullying and depression. Depression is influenced by many factors: social, environmental, genetic, but our findings suggest that variations in TPH1 may shape how sensitive young people are to their social experiences, including bullying. This is only one piece of the puzzle when trying to understand how 2 kids exposed to the same social environment might have very different outcomes. It is also important to acknowledge limitations of our current study. Our sample size was modest, and most participants were of a similar background. We need larger and more diverse groups in the future to confirm these findings. As well, depression was measured at a single time point, and we know mental health changes over the years. In the future, we're hoping to examine different types of bullying, whether that's physical, verbal, cyber, relational, and taking into account scores we have for multiple time points as participants grew up. We also hope to continue exploring gender differences and expand to include more diverse populations. Ultimately, this study is a big step in offering early evidence that genes involved in serotonin production, like TPH1, may help explain why some youth are more vulnerable to depression after being bullied. Understanding these interactions pushes future research that will increase our ability to identify at-risk youth and create targeted, preventative, and personalized treatments. Bullying is incredibly common, and research like this works towards solutions that consider not just the environment, but also unique biology of each individual. Here are my references, and thank you so much for listening.